I am Dr. M.S.V. Chalam, and today I would like to give a brief presentation on Chaetotaxy in identification of Lepidopteran caterpillars. Next slide, please. So we all know that Lepidoptera is one of the most important order, second largest order. The students must have studied in their taxonomy course. Around 1,60,000 named species are there, including butterflies and moths. But economically, this is one of the most important order because majority of the crop pests are included in this order. Next, please. So my talk will be on the below contents: introduction, importance of chaetotaxy, classification of chaetotaxy, steps in conducting the research, a few examples of chaetotaxy of different caterpillars, role of chaetotaxy in taxonomic identification, as well as conclusions and future prospects. Next, please. So you see, the larval stages of Lepidopterans, they play a very crucial role in crop losses. As we all know, the Lepidopteran pests, they will be devastating a major field as well as the horticultural crops. And the majority of the Lepidopteran caterpillars, they will be feeding on almost all parts of the plants like foliage, bud, blossoms, roots, whatnot, and everything, causing a considerable crop loss. And we see the before proceeding for any of the management strategies, before applying any of the IPM strategies, the knowledge on accurate identification of the pest is always important in order to formulate the effective management strategies. Next, please. It is very easy to identify the Lepidopteran adults, maybe moths or butterflies. We do have subtle identification features, specific marks of identification with which we can identify the adults. But the main damage is caused by the immature stages, that is the larval stages, where the identification of these immature stages poses considerable difficulty because majority of the caterpillars, they may be similar, they may be existing in various color morphs. Sometimes it is very difficult to identify the Lepidopteran caterpillars, especially damaging the crops. Next, please. So if you see what is the role of the, but before going to the structure, let me tell what is chaetotaxy. You know, the study of arrangement of bristles on the seta or bristles on the larval body is nothing but the chaetotaxy. How this particular study of larva is, or else the chaetotaxy is useful for the identification. We know the immature stages of Lepidopterans are called as caterpillars, and they are usually eruciform with a well-developed head and a cylindrical body. You could able to see the caterpillar here. Next, please. So this is the general structure of the larva. Next one, please. So coming to the various morphological features which we will be using in the chaetotaxy. First one is the antenna. In case of the Lepidopteran caterpillars, the antennae are short, much reduced with bristle-like in appearance, three-segmented, strong mandibulated mouthparts. Ocelli are generally referred as stemmata, and there are 6 ocelli arranged in circular or semicircular fashion. Compound eyes are absent. Next one. So here we will be on head. What are all the characters we will be useful for that will be useful for chaetotaxy? Generally, majority of the caterpillars, they will be having a well-defined sclerotized, deeply pigmented head, and on the head we will be observing epicranial suture, that is inverted Y-shaped suture. There are 2 peculiar sclerites. These sclerites are called as head frontal sclerites, which are existing on either side of the facial sclerite, though that is the frons or frontal sclerites. These Two sclerites will be having a taxonomic importance in the identification. Apart from that, you will be observing the clypeus as well as the labrum, and the stomata are present. The number of stomata always varies. They may be from 1 to 6, which are located on the lateral aspects of the head, and we will be observing a distant protruding spinneret. Next slide, please. See, before understanding the chaetotaxy, there are certain terms which we must understand. So, the Lepidopteran caterpillar will be having several armature on its body. So, what is here armature? The seta, the spines, the pigmented areas, the enlarged areas or the round areas, the stripes, spots, all are referred with a common name, which are called as armature. So the different armature which we will be using in the description of a caterpillar are as follows. First one is a seta. A seta is a hair-like organ arising from a ring-like papilla and is connected at least with one hypodermal cell. That particular hair-like structure is called as seta. Next one is pinaculum. So what is this pinaculum means? A flattened, heavily pigmented, sclerotized area occurring at the base of the seta is known as pinaculum. Next one is chalaza. If in the a cone-like elevated structure present on the pinaculum is called as chalaza. Next, on the chalaza, we will be observing several spines. If it is a single spine, we will be generally referring it as spine. And if there are several spines, we will be referring it as spinules. A structure with several of the spinules is called as scolus. If the chalaza is bare without any seta or spines or spinules, it is called as cornicula. And if the cornicula is raised, it is called as tubercle. So, a seta or pinaculum or chalaza or spinules, scolus, cornicula, these are various morphological terms useful for describing the armature, that is the general morphological structure.
Yeah, excuse me, Dr. Chalam.
Yeah,
I request your camera should be on so that,
yes.
because the Dean SPG's office is recording. So kindly switch on your camera, please.
Yes, sir. Now it is on, sir.
Oh, okay. Thank you.
Thank you, sir. Next slide, please. You see, okay, apart from that, there are other terms. First one is veruca. So what is veruca? If the seta are numerous, which are arising from a round wart-like elevation, this is called as veruca. The another term is vericule. What is the difference between the veruca and vericule? If the seta are present in a thick group, for example, if we see the hairy caterpillars, orchidids as well as lymanthids, we will be observing the spines which are more or less parallel to one another. Such type of structure is called as vericule. In some caterpillars, we will be observing some swellings present on the dorsal side of the body. The enlarged dorsal swellings are called as gibbosity. If the swellings are without definite outlines, these are called as protuberances. Next, if you observe the caterpillars under high magnifications, so small spine-like structures are observed, which are called as microspines. So the major difference between the seta are macroscopic, you can visualize with your naked eye, whereas the microspines cannot be visualized with your naked eye. We require a high-end magnification. Next, please. Next slide, please. Yeah. So this one is pinaculum, verruca, as well as simple seta, verruca, and verricule. You could able to see verricule means there will be a lot of parallel seta or parallel vines. Just like the verruca means it will be just like our pin cushion. Next one, please. So these are different. Absolutely, there is no need of explanation. There are several. These are some of the Lepidopteran caterpillars like semilopers, lopers, hornworms, hairy caterpillar, slugs, bagworm. There are n number of caterpillar various morphs of caterpillars present in the order Lepidoptera. Next one, please. So if you see the leg segment, again the caterpillar will be having a true legs as well as the prolegs. Prolegs are present on the prolegs will be having a soft fleshy tissue called as planta. On the soft fleshy tissue, we will be observing several of tiny hooks. These hooks are called as crotchets. Next one, please. Next, we will be going for definite chaetotaxy. Next one. So what is chaetotaxy? So till now we have seen what are the different various morphological terms which are used in description of the chaetotaxy. Now we will be knowing so what is meant by actual chaetotaxy and how to read it. So insect chaetotaxy is nothing but a study of arrangement, number of types as well as the seta on various parts of the body. Generally, the seta are observed on the head, thorax as well as on the abdomen. The arrangement of seta is very, very specific to a particular species or to a particular family. And for study of this particular chaetotaxy, we require some specialized equipment, maybe microscopes or even SEM. Next one, please. So generally, insect chaetotaxy is useful for the identification as well as classification of the insects, and chaetotaxy also provide insights on evolutionary relationship between the different group of insects, because the pattern of the seta can indicate a common ancestry. Therefore, the in-depth study of the chaetotaxy can solve certain of the even the phylogenetic problems. Next one, please. Yeah. Now you see the first one is based on crotchets. How to study the crotchets? We all know on the prolex we will be observing a soft pad-like cushiony structure which is referred as the planta. On planta we will be observing several of the hook-like structures which are called as crotchets. These crotchets may be arranged in different circles. For example, the caterpillar is having only one circle or of crotchets, one series of crotchets. The number of circles is referred with the term serial. Uniserial means one series. Biserial means we will be observing two series of crotchets. Multiserial, more than two, maybe 3, 4, 5, whatever the case may be, we will be referring with a simple term, that is a multiserial. So number of circles are referred as a serial, uniserial, biserial as well as the multiserial. Next one, please. So even just like the number of circles, the length of the crotchets will also be varying. For example, uniordinal. If all the crotchets are of a single length, it is called as a uniordinal. If the crotchets are of two different lengths, it is a biordinal. If the crotchets are of three different lengths, it is a triordinal. If the crotchets are of more than three different lengths, we will be using multiordinal. So while description of the crotchets, we will be calling it as uniserial, uniordinal crotchets. One series, one length. Uniserial, biordinal, uniserial, triordinal, uniserial, multiordinal. Like that, biserial, uniordinal, biserial, biordinal, biserial, triordinal, biserial, multiordinal. We can have the combination. So series refers to the number of circlets, whereas ordinal, the term ordinal refers to the length of the crotchets. Next one, please. But sometimes it may so happens that the circlet is incomplete. We will not be having a full circlet. In such cases, it is called as penilips, that is incomplete circle of hooks or crotchets is referred as penilips. Next one. The penilips is again, if it is a dorsal side, if the break is on dorsal side, it is called as a dorsal penilips. If the break is on ventral side, it is called as a ventral penilips. If the break is on mesal side, it is a mesal penilips. So we will be again, if you see uniordinal, uniordinal dorsal penilips, uniserial biordinal ventral penilips. Like that, we will be having n number of combination. Next one, please. So you see the arrangement and well as the nomenclature of the seta present on the body are most important. Next one, please. Next one, next slide, please. You see, now what is a setal map? So through Chaetotaxy, we will be constructing the setal maps. A setal map is nothing but the arrangement of seta primarily present on thorax as well as the abdomen. A setal map depicts the arrangement of seta on one half, maybe on the left half, maybe on the right half, one half of any of the thoracic or abdominal segment. Again the seta of the caterpillars can be divisible into three types. First one are the primary seta, subprimary seta and secondary seta. Generally, primary seta are found in the first instar and are constant throughout the larval life. Once the first instar larva acquires the primary seta, their position is constant and they will never be changing even though the insect mold. For example, we can compare the primary seta with our own birthmarks. We will be having our birthmarks and they are constant throughout our life. Like that, the primary seta, they will not be changed throughout the life of the larva, and even if the molt, they are not lost. Next. Subprimary seta previous slide please, previous slide please. The subprimary seta, they will be appearing after first molt, that is during the second instar larva. Just like the primary seta, the position of the subprimary seta is also constant throughout the larva. Next, the secondary seta, generally they will be appearing only after the second instar, mainly during the third instar, and their position is not constant and even they bear no significance in the Chaetotaxy. So several of the students, they will be putting one question, sir, which instar we have to choose for studying of the Chaetotaxy? Primarily we should choose third instar because the primary seta will be appearing in the first instar, subprimary seta will be appearing in the second instar. Both they will be having a definite pattern and will be retained throughout the life of the larval stage. Therefore, we have to definitely take third instar for study of the larval Chaetotaxy. Next one, please. So each seta is given with a specific name. For example, those seta which are present on the prothoracic shield, they should be referred with the word abbreviated as XD, XD1, XD2. Dorsal seta, they are referred as D1, D2. Subdorsal seta, they are referred as SD1, SD2. Lateral seta, they are referred as L1, L2, L3. Subventral seta referred as SV1, SV2, SV3. So by seeing the position of primary and secondary seta, they are highly constant. They are highly conservative. Absolutely, there will be no change with respect to the larva or with respect to the individual. So with these ones, that is XD1, XD2, the descriptions D1 or D2, we have to describe or we have to position the seta. Next one, please. So how to conduct research on Chaetotaxy? Probably all the students might be conducting research on any one of the Lepidopteran caterpillars. So this is a very easy the study of Chaetotaxy for a particular Lepidopteran caterpillar is very easy. It won't take more than 1 week if you have your caterpillar population ready. You can incorporate this as one of your objective, maybe for your MSc or for your PhD research. So how to conduct the research? These are basic prerequisites: collection of the larva, killing of the larva, preservation. Sometimes you need to go for rearing, mounting. The last one is photographing or construction of the statal maps. Next one, please. So you see, there are many techniques. Absolutely, at this stage, we don't need the techniques for collection. You can visually observe the crops. You can observe for the damage symptoms. You can observe for the feces, and immediately by visual observations, you could able to collect the larva. Next one, please. So, for example, you can use several of the nets, sweep nets, beating sheets, funnel traps, several nets, as well as by any means you can collect the caterpillars. Next one, please. So even you can collect the egg mosses, and the egg mosses can be reared in the laboratory in order to obtain the caterpillars. But the rearing is a little bit difficult. You require specific conditions, maybe temperature, humidity, like that. Next one, please. Okay, you have collected the larva. How to kill? There is a one mixer by name KADD mixer that is prepared by mixing kerosene 1 part, 95% ethyl alcohol 7 parts, dioxin 1 part, and glacial acetic acid 2 parts. This is called as KADD mixer. If kerosene is not available, just you can call it as a AAD mixer. You can mix ethyl alcohol, dioxin, and glacial acetic acid. Why? Because even you can use formalin for killing of the Lepidopteran larvae, but only thing is it will distort the color. The colors will be faded away if you kill with the if you dip the caterpillars in the formalin, and it will be causing the shrinkage of the body parts or the segments. In such cases, it will become very difficult to read out to note the Chaetotaxy. Therefore, the KADD or ADD mixture is a satisfactory killing agent for killing of the Lepidopteran larvae. Next one. Immediately after killing, you can insert the kill larvae, the dead larvae, in the potassium hydroxide, then pass it through different grades of alcohol, 60, 80, 95%. Each grade, you can retain the insect specimen for about 10 to 15 minutes in order to facilitate the dehydration. We will be using potassium hydroxide to macerate the tissues and to remove the unwanted tissue. Next one, please. So the digested soft tissues are removed with the help of a blunt needles, and the specimens should be transferred to clove oil for clearing, and you have to retain these specimens in phenol xylol mixture 1:1 ratio until you can spread the insects. You can spread the insects on slides and mount them either with the Canada balsam or else DPX mountant. Next one, please. So if you want to preserve, here you see you can conduct the Chaetotaxy studies, or else you can go for molecular characterization of these caterpillars by simply preserving them in 95% of the ethyl alcohol. So if you want to conduct your Chaetotaxy studies at a later date, you have collected in the field, you have killed it, and if you want to store them for few days or few weeks, let it be stored in 95% ethyl alcohol. Never store it in formalin, because we will be observing the distortion of the specimen. Next one, please. So here there is one important part is rearing. Why we have to go for rearing? Generally, Chaetotaxy can be done on field collected specimen also, but we are not sure of identification of a Lepidopteran caterpillar based on the larval characters. Generally, the Lepidopteran taxonomy is mostly based on the adult characters, especially the male and female genital characters. We have to dissect out the male and female genital organs. We have to observe the characters. Then only you can confidently say to which particular species it belongs. But if you rely heavily on the larva, and if you do not study the adult character, sometimes because of the color variation or color morphs, your identification features may go wrong. Therefore, a perfect chetotaxis study involves the rearing of the larva in the laboratory, obtaining of the adults, sexing of the adults, mating of the adults, then conduct the chetotaxis studies on the progeny, and before conducting the chetotaxis studies on the progeny, get confirmation of the adults from a Lepidopteran taxonomist, so that you are 100% sure that your chetotaxis studies will never be going wrong. So for such cases, we have to rear the caterpillars. Next one. But if you generally, if we go for one or two study of the chetotaxy in one or two caterpillars, it will be very easy for you to rear the caterpillars. But if you are experimenting on, if you are exclusively working on chetotaxy of a number of caterpillars, it becomes very difficult, because the larvae are they require specific food, and they require specific temperature and environmental conditions. In such cases, if you go for number of chetotaxis studies on different number of larvae, you may face a little bit difficulty. Next one. So after the chetotaxis studies, you have to take the photographs, normal photographic equipment attached to our Olympus, anyone, trinocular stereo zoom microscope. Nowadays the facility is available everywhere, and you can take the beautiful photographs. Next one, please. Next, even you can conduct the morphometric studies. Chetotaxis studies are always associated with the morphometric studies. You can take the width of the head, length of n number of morphometric studies you can conduct on the collected caterpillars. Next one, please. Next, few examples I will be giving. So you see, this is the chetotaxis studies conducted on Spodoptera littora. First one is the head capsule. You could able to clearly see the inverted Y-shaped head capsule. The B is the larva, C is the crotchets. On the right side, you could able to see this is the camera lucida diagram of pencil drawn diagram of thorax as well as the abdomen of the caterpillar. Next one, please. So if you see a brief description, how you will be describing. Next slide, please. So here you see the description about the caterpillars. It is having dorsal, subdorsal, supraspiracular, subspiracular stripes, head projected ventral side with an inverted Y-shaped suture, and the most important one is chetotaxy. You see the spines. Prothoracic shield is chitinized, extending up to the margin of XD2. XD2 means it is the dorsal setae. Dorsal setae XD1, XD2. The subdorsal setae D1, D2 are present. Lateral setae L1, L2 are present. So you will be giving the, if you observe under the microscope, you can draw the camera lucida diagram, and you can simply number the theta based on the nomenclature, and you can describe the thorax as well as the abdomen. Next one, please. So like that, generally we will be describing the chetotaxy of the thorax as well as the third abdominal segment. So why to choose for the third abdominal segment? Why? Because generally we will be detaching the thorax as well as the abdomen for conducting the chetotaxy studies of thorax. So if you take first abdominal segment, it may so happen that while detaching the thorax and the abdomen, there is a likelihood chance that first abdominal segment may get damaged. In such cases, in order to prevent this one, we will be leaving the first and second abdominal segment, and we will be conducting. Here also you see dorsal seta, the position of the dorsal seta, subdorsal seta, lateral seta, ventral seta. Like that, we will be describing the chetotaxy of thorax as well as on the abdominal segment, and we will be describing the arrangement of crotchet. It is a uni-ordinal mesoceres. So here also, if you want, you can have a description of adults also. Next slide, please. So this is the first one is the Chaetotaxy of Spodoptera litera. This is the Chaetotaxy of Spodoptera frugiparda. I'm not going to go into the entire details. Here also we will be describing the shields as well as the seta which are present XD1, XD2, dorsal seta D1, D2 like that. Next one. Here now because of the you could able to clearly see the arrangement of crotchets. You can take beautiful photographs of the crotchets as well as you see E, the Chaetotaxy of thoracic sequence. Quite evident. Sometimes you may get very difficult. You may get confused in the identification of caterpillar. For example, Spodoptera litera, Spodoptera frugiparda. But the position of the seta is very much specific, completely different from that of litera as well as the frugiparda, and by conducting the Chaetotaxy studies you could be able to clearly differentiate or distinguish between them. Next one, please. So this is the Chaetotaxy studies on Helicoverpa armigera. I can show n number, but the time is very much limited. We can that you can go for the description. So here my main intention is that I am showing only the three caterpillars: Spodoptera litera, frugiparda, as well as Helicoverpa. Maybe you may be working with Nephelocrasis medinalis. You may be working with Scirpophaga incertalis. You may be working with another caterpillar. So you can incorporate one of the description, the Chaetotaxy description as your objective, and you can simply describe. It won't take much time, and moreover, it adds to the Chaetotaxy database. Next one, please. You see the Chaetotaxy of prothorax, mesothorax, Chaetotaxy, the position of the seta. Next one, please. This is about the spotted pod borer Marca vitrata. Next one, please. So, how the role of what is the role of Chaetotaxy in the taxonomic identification? First one is the study of Chaetotaxy is useful for species differentiation because you see the presence, absence. We have not gone into the in detail of a particular. Even if you study the setal number, the setal position will be varying in between the species. You may conduct the Chaetotaxy studies for Spodoptera litera, Spodoptera frugiparda, Spodoptera exigua, Spodoptera mauritiana. All the four species will be having a different setal maps. All the four species will be having different crotchet arrangements. So you could be able to easily differentiate between the species. Even by studying the Chaetotaxy, we can differentiate between two closely related species that may appear very similar based on the morphological characters. Next one, please. So, the studies of Chaetotaxy are useful for the character variation. You could able to construct an identification key. So, probably in your MSc or PhD, you must have studied different types of key. We will be using simple bracket key or simple non-bracket key. With the help of the Chaetotaxy, with the help of the explored larval characters, you can simply construct a pictorial key for the identification of the caterpillars, which will be very useful for the field level workers, may not be for the entomologist, but for the field level extension worker. By simply studying the characters, the color, the banding pattern, they could able to easily identify. So the Chaetotaxy studies are useful for construction of taxonomic keys. Next one, please. Grouping, taxonomic grouping. So species which are having similar Chaetotaxy studies, maybe at the family, genus, or maybe at higher taxonomic levels, may be grouped, which will be aiding for broader classification of the organisms. Next one, please. So ecological adaptation, of course, this is a very minor one. The presence or absence, or else the position of seta on specific body parts can be correlated with the habits, habitats, lifestyle as well as the feeding habits of the organism. Thereby, we will be knowing the ecological adaptations of that particular caterpillar. Next one, please. Evolutionary study. So, the comparison of setal pattern, that is nothing but a setal maps. The caterpillars which are having similar setal maps, you can say that they may be having a common phylogenetic ancestry. That means you could able to conduct evolutionary studies and you could able to assess the morphological diversities through the study of Chaetotaxy. Next one, please. Systematics. It is the most important. The five are not so important from the study of from the systematics point of view. Lepidopteran systematics is mostly based on the adult characters, but not on the larval Chaetotaxy. So maybe some of you may be studying the Lepidopteran taxonomy. While studying the Lepidopteran taxonomy, please include the larval Chaetotaxy as one of the objectives, because the larval Chaetotaxy also useful for establishment of the taxonomic relationship and contributes for a greater development of insect biodiversity. If you combine the insect biodiversity, the Lepidopteran taxonomy as well as with the larval Chaetotaxy, then only the systematics of Lepidoptera will be complete. Otherwise, there will be some lacuna if you study only the adult genital characters without studying the Chaetotaxy. Next one, please. Species. Generally, those Lepidopteran taxonomists which are describing a new species, they will be mainly concentrating upon the adult characters. But if you incorporate the Chaetotaxy studies also, this information is especially useful for the subsequent researchers who could able to identify maybe the larval cat, maybe the immature stage larval characters, maybe the adult one. Next one, please. So, nearing to the end, what are the conclusions? Chaetotaxy plays a pivotal role in the identification and the classification of the larvae, and the arrangement of these seta is very species specific, and it can aid in helping the taxonomist in differentiating between closely related species. And Chaetotaxy patterns can be serving as diagnostic tools in pest management as well as monitoring programs, and even you could able to rely on Chaetotaxy for early detection of pests and implementation of targeted control programs. Next one, please. So, future prospects. How we can deal it in? You see, now for study of Chaetotaxy, we always require imaging software, but we are having very good imaging softwares with very good microscopes. Therefore, with the advent of new microscopes, with the advent of high resolution microscopy, the Chaetotaxy patterns can be easily studied. Absolutely, there is no need to go for old cameralosa diagrams. You can directly take out the photographs, which improves the precision of taxonomic identification. Next, development of a database on Chaetotaxy. We are not having any database on the Lepidopteran Chaetotaxy. Just if you study the Chaetotaxy and a database is developed maybe at the university level, that goes a long way. And the efforts of these taxonomists can on the Chaetotaxy will be having a holistic understanding of the ecological and evolutionary implication of the insect larvae. Next one, please. So, thank you for patient listening. Any questions or doubts?
Dr. Chalam, thank you very much. So
Thank
now
you.
I request our students, young and energetic, even after hungry, they are very.
Yes, sir. Yes, sir. It is
Yeah, they are very eager to ask some questions.
yeah, definitely, sir. Please, sir.
Yeah.
Please.
So Nitin, let us start from Nitin. Come. You ask some questions, Nitin. It's your syllabus also. So sir
One of
has
our
covered. Actually, today you have covered on syllabus for the students. Thank you.
yes, sir.
So it is
Thank
a master's lecture, one lecture. So it will
maybe,
be
sir.
fine if.
One of our student Muthulakshmi may be present, sir. I don't know
Yeah,
whether
yeah,
she
yeah.
will.
So she is,
Muthulakshmi Bhavitra, she did our MSc at Tirupati, sir. Yeah. I could able to see her. Yeah,
Okay,
fine.
fine.
Thank you,
But
sir.
actually, she tried to dodge your lecture. I only brought her inside. Oh,
She might have heard in her MSc, sir. That's why. Yeah, please, sir.
Chaetotaxy studies in flies at all, sir.
Yeah, actually, Chaetotaxy is well studied only in the two orders. First one is the order Lepidoptera, second one is the order Diptera. In Diptera also there are several Chaetotaxy studies based on the arrangement and position of bristles. I haven't touched because that itself is another topic. We can identify both Diptera as well as Lepidoptera based on Chaetotaxy studies. If you are working on Diptera, yes, please go for Chaetotaxy studies on.
Good afternoon, sir.
Good afternoon.
Sir, my question is about in case of Lepidoptera, it's a very large insect, so we can easily find out the chaetotaxy. Nitin told in Diptera how to find the chaetotaxy because it's a very small insect also.
See,
It's very difficult totally no.
Dipteran chaetotaxy is not based on the maggots. It's based on the bristles which are present on the thorax. You can easily study this chaetotaxy under a microscope. For example, I will give you one example. The entire taxonomic separation of leafhoppers is based only on male genitalia. So we are studying male genitalia, especially I am specialized in leafhoppers and plant hoppers. They are not at all visible for the naked eye. Only under a microscope you could able to see. Otherwise, no. So you can study the Dipteran chaetotaxy, but you require a specific microscope. Especially you have to work on thorax as well as the abdomen of adult flies. Then you could able to easily. Good morning, please.
Sir, you told that in preservation we can use 95% ethyl alcohol.
Yes.
How long we can preserve with that, sir, larva?
Actually, I have preserved up to 6 months. Absolutely, there is no problem with that. But only thing is that lid should be tight enough. That's all. Up to 6 months, absolutely no problem. Within 6 months, you could able to conduct your studies, isn't it? Up to 6 months, if you store it in ethyl alcohol, your specimen will be as fresh as newly collected. Good afternoon.
Myself Subhashri from First MS Entomology, sir.
Yes, please.
Sir, your lecture was very much useful, sir. Thank you for the lecture, sir.
Thank you.
So my question is, sir, is there any chance the chaetotaxy must have changed during the process of evolution, sir?
No. Chaetotaxy is always constant. For example, if you want to study the phylogenetic relationships, we will be depending on the chaetotaxy also. It will never change because the position of theta are so constant that while evolving the particular species, the position of the theta, primary and subprimary theta are so fixed that it will never change unless there is a new species formation.
Yes, sir.
So I think you could able to understand.
Yes,
within that particular species, the position is constant. But if there is a new species, definitely the position will be varying.
Very good afternoon, sir.
Good afternoon.
So I just want to know whether this chaetotaxy study can be applied for hairy caterpillars, sir. Is it possible or?
Definitely you can do the chaetotaxy studies, but it will be a little messy. Why? Because we have to work with a number of hairs. You see, probably you might have seen the fourth or fifth instar of a hairy caterpillar and you may be confused, sir, there will be so many hairs, how to study this one. But the position of hair, we have to take only the newly hatched third instar larva where the position of hairs will be very minimal. We can definitely go for the, even for hairy caterpillars, the chaetotaxy studies. It will be very easy, but only thing is number of hairs will be more. You have to work a lot when compared with the other categories.
Taxonomy for my PhD thesis. I want to ask, sir, is there any consolidated database where we can access information on different chaetotaxy of different Lepidopterans?
Yeah, unfortunately, till now, there is no database on this chaetotaxy, just like a genomic database. We are not having any database because the Lepidopteran taxonomists have mostly relied on the adult characters, not on the chaetotaxy. Therefore, I'm very sorry to say that absolutely there is no database. We have to develop. That is a future area where we have to develop the chaetotaxy database.
Afternoon, sir.
Good afternoon.
Sir, is there any chances like for student or for a graduate we can identify, but is there any formal level identification of the larva in the
Yes.
formal level?
Yeah. So we can develop a pictorial key. The Chaetotaxy study is only for the entomologists, not for the farmers. So while studying the Chaetotaxy, what you can do is you can entirely describe the larva along with good quality photographs, which can be useful for a ready reckoner for the farmer for the identification. We can construct a pictorial key which will be useful either for the farmers or even for the field level extension workers. We can combine the pictorial key as well as the statal maps so that this particular key can be used at the both by the farmers, the field level extension workers as well as the entomologists who are working for a specific crops. But for farmer, no, the Chaetotaxy is not useful because he may not understand.
Sir, and is the in the CTs are present in any pupil stage? So pupil.
No. The Chaetotaxy study in case of Lepidoptera is only restricted to the larva, not for the pupa. We will not be observing any definite pattern of, we will be observing marks, certain small hairs, but there is no definite pattern for this pupa. We cannot rely on the pupa, the hairs or hairs present on the pupa for construction of the settle map. It is only on larva.